Hi there! You may think that I'm going to tell you something about the weather today. No, sorry. We are going to talk about epigenetics. How exciting is that? Oh, and why I did bring this? I will explain that to you later. To understand the concept of epigenetics in relation to cancer, it is essential to have some basic knowledge about cancer biology. Cancer is caused by changes in the genetic material of our cells. The DNA sequence contains clues, the genes, for making proteins that carry out the function of a particular cell. Changes in the sequence of the DNA, called mutations, can result in the development of cancer. For instance, if the DNA sequence of a gene responsible for cell death changes, this gene may become inactivated. As a result, cells will not die. Cell growth continues in an uncontrolled way and the tumor develops. There are also other changes to the DNA that can contribute to cancer development. You can think of folding or marks on the DNA. This is called epigenetics. Epigenetics is another way of controlling the activity of genes. Let's see why this is relevant. We know that all our cells have exactly the same DNA sequence, but of course, different cells have different functions. I will give you an example. We have a heart cell and a skin cell. Their DNA sequence is identical, but their function is different. Each cell is specialized to perform a function in your body. How does this work? It has to do with turning genes off and on. Switching genes off or on is called gene expression. An expressed gene is active, meaning that the protein is formed. The folding and marks on the DNA that I mentioned earlier can determine whether a gene is expressed or not. One of the marks on the DNA mostly studied in relation to cancer is called DNA methylation. DNA methylation occurs by the addition of a methyl group to the DNA. These methyl groups act as signals along the DNA and can help to determine whether a gene is expressed. Methyl groups are available from biochemical reactions in our body and are also strongly dependent on dietary factors. An overload of methyl groups is called hypermethylation. Hypermethylation of important regions of genes, so-called promoter regions, can inhibit gene expression. Let me explain how we think this works. Imagine that the methyl groups are represented by umbrellas, while the genes are the flowers in your garden. With many umbrellas, no sun and water will reach the flowers and they will not grow. Likewise, if there are many methyl groups in the promoter region of a gene, the factors needed for gene expression cannot bind and the gene is switched off. As a result, no protein is formed. In cancer, we often see that important genes are switched off due to an overload of methyl groups in the promoter region of these genes. This can occur, for example, in DNA repair genes, which are essential for repair of errors in the DNA. As a result, DNA damage will not be efficiently repaired, which may lead to further mutations and, in the end, uncontrolled cell growth. On the other hand, if there are few methyl groups, or umbrellas if you'd like, this may also have serious consequences. A lack of methyl groups is called hypomethylation and can lead to a reduced stability of the DNA. And yes, this has been linked to an increased cancer risk. So of course, I did not talk about the weather. What I have tried is make you better understand how DNA methylation regulates gene expression by using the example of my umbrella. And more importantly, why epigenetics can be relevant for cancer development. Oh, I better go now. See you.